welcome you to guest lecture under 3-in-1 program 2020. And the topic of guest lecture today is understanding the basic properties of enzyme in fermentation process, organized by Department of Agricultural Product Technology. Head of Department of our Agriculture Product Technology, Universitas Brawijaya. I would like to say thank you very much for Profi coming here and join in this guest lecture. And before we start the guest lecture, I will probably he was graduated PhD from Tokyo University, and he got a professor on 2018, Sensei?
I think so, yes.
And he is also one of the lecturer of my PhD during my course. He will give a talk for food biotechnology program. So, for Professor Yang, please start your presentation.
All right. Thank you, Koris-san. Can I share my file, PowerPoint file? Can I share?
Yes.
Okay. So let me see. Okay, good morning, everybody. I am Toshiharu Yakushi from Yamaguchi University, Japan. So I'd like to talk about, let's see, fermentation and enzyme. So I like to use a PowerPoint file. So let me share my file. これか。Okay, today I'm going to, can you hear me?
Yes, Sensei.
Okay. So I'm going to talk about basic properties of the enzymes and in fermentation process. So, first of all, where am I? So maybe you are here. Okay, I think so. So it's just 2 times time difference between us, right? I'm here. And you know, Japan consists of 1, 2, 3, 4 main islands, big islands. So this is the main island. And I'm in Yamaguchi here. So you can see the Japan here, then I'm here. And maybe you know Tokyo is a capital city in Japan. Tokyo here. So I hope next year we will have Olympic in Tokyo. But I'm not sure we can do it or not. So anyhow, here is Tokyo, and here is Kyoto, another famous city, a historical city in Japan. And this is a nice pagoda in our place, very near. I think we can go there maybe 10 minutes drive or 15 minutes by bike. Yeah, we have such a nice pagoda near here. So let's start my talk today. So today we will discuss 3 questions actually. So what is the first one is what is fermentation and what is enzyme. And finally, we will discuss the key factor of enzyme in fermentation. So let's start. So what is fermentation? So actually, it's just a definition, and I checked Wikipedia recently. So Wikipedia said fermentation is a metabolic process that produces chemical changes in organic substrates. Organic substrates through the action of enzymes. This is a definition in Wikipedia. So it looks, for me, it looks strange. So fermentation is a chemical change through action of enzymes. It little bit strange for me. So what is strange for me? So for example, let's consider malt. Malt. Do you know malt? So malt is germinated grain. So usually wheat. So malt is germinated wheat grain. So let's consider, if you consider malt, it's just same as this. I mean, malt is germinated wheat grain, and how do I say? It's induced chemical changes in wheat grain. Actually, chemical changes wheat grain through the action of enzymes. So just same as definition of fermentation in Wikipedia. But we don't think, we don't consider malt is fermentation. We don't think, we don't consider, we consider malt is not fermentation. Okay, malt is not fermentation. So if I say, I think a fermentation is actually chemical changes in foods, maybe usually foods, with the aid of microorganisms. That's my definition for fermentation. So therefore, malt is not fermentation product because malt is not involved in microorganisms, just germinated wheat grain. So that's my definition for fermentation. And what is the chemical changes? So maybe flavor or taste. I think the flavor and taste are very similar to each other actually. And another important chemical change is maybe nutritional properties. So if it's, how do I say, difficult to degrade by our body, in that case, such a microorganism function may help to degradation or intake to our body. The changes may be in nutritional properties. And another important chemical change may be color. It looks good. That induce our, how do I say, our patient, our thing to have a food, actually. So chemical changes includes many functions of foods, actually, flavor, nutritional properties, and color. So microorganisms change such things. But I like to put some additional information to this definition, actually. So finally, I like to define the fermentation like this. Fermentation is chemical changes in foods with the aid of microorganisms, which are beneficial to humans. So it should always it's just storage or decomposition. So it's like chemical changes in foods with the aid of microorganisms, which are not beneficial to human. It's just storage. That's different from fermentation. Fermentation is always good for humans. Okay, that's my definition of fermentation. So next, I'd like to talk about enzyme. What is enzyme? So can anyone answer my question? What is enzyme? Please raise your hand. What is enzyme? Who can answer? No?
Shy.
Kore-san, can you answer?
Yes, enzyme is a protein that has a catalytic site and can convert from the substrate into the products.
Good. Very good. Yeah. Actually, I check Wikipedia again for enzyme. And Wikipedia say enzymes are proteins that acts as biological catalysis or biocatalysts. So maybe good. This is true, I think. I agree with Wikipedia definition. And also, Kore-san, Kore-san said also proteins. Enzymes are proteins. Yes, that's right. But we know some exceptions. Actually, only one exception. So therefore, I like to say I don't like to say enzymes are proteins because there is an exception. So just I like to say enzymes are biocatalysis. I like to say this. So what is exception? RNA. So I think several enzymes made of RNA. There are several enzymes made of RNA actually, but I think most important or most famous RNA enzyme or ribozymes is ribosome. So, you know, ribosome is very, very important in our body because it's protein synthesis. It's important. It's actually the site of protein synthesis, very important enzyme. So ribosome, catalysis of ribosome is RNA. So therefore, we should include RNA in enzymes. Okay, and yeah, enzymes are biocatalysis. And catalysis, what is catalysis? So catalysis accelerates chemical reactions. So this is a general definition of catalysis. And enzymes. In case of enzyme also accelerate chemical reactions, but it's limited in biological process. Okay, so enzymes accelerate chemical reactions related to biological processes. So we call this such chemical reactions related to biological processes. We call it metabolism. So if you think about enzyme, that means we think about metabolism, chemical reaction in our body, or chemical reactions related to biological processes. So my definition of enzyme look like this. Enzymes are biocatalysis which accelerate chemical reactions related to biological processes. Okay, so that's the definition of enzyme. My definition, actually. So next topic is what is the key of enzyme in fermentation? So of course, catalysis, that's very important in enzyme actually. So if you think about what's this? Lactic acid fermentation. So lactic acid fermentation, you can see this fermentation in yogurt. Maybe my pronunciation is not good, but it's fermented milk. You can see lactic acid in many places. So this is basically just same as our glycolysis, actually. So this is a cartoon metabolic pathway of glycolysis. This is start. Glucose can be changed by a series of chemical reactions. And finally, we will get pyruvate. That's glycolysis. This is the same as our glycolysis in our body, glucose to pyruvate. Just same as glycolysis in our body. But actually, the real lactic acid fermentation is different, just a little bit different from our glycolysis. But a basic concept of lactic acid fermentation is very similar to our glycolysis, actually. So if you produce pyruvate from glucose, pyruvate just reduce to lactic acid by a specific enzyme, lactate dehydrogenase. So this is a really important enzyme in lactic acid fermentation. if you don't have this enzyme, you cannot produce lactic acid actually. So this is just simple reduction of pyruvate. You can see keto group here. So this keto group is reduced to lactic acid like this. Then we will get glucose. We will get 2 lactic acid from glucose. That's lactic acid fermentation and lactate dehydrogenase. So if you think lactate dehydrogenase to produce lactate from pyruvate, so that's a chemical reaction catalyzed by lactate dehydrogenase. The reaction like this. So if you, in this case, reduction of pyruvate, if you want to get lactate, you need to reduce pyruvate. Lactate dehydrogenase is a member of oxidoreductase. So we can, how do I say, we can categorize enzyme into 6 group. Then one of group is oxidoreductase. This enzyme catalyzes oxidation and reduction of compound. So lactate dehydrogenase is, this means lactate dehydrogenase means lactate oxidation. This enzyme catalyzes lactate oxidation. Actually, you can consider the reverse reaction of this reaction. If you think reverse reaction of this chemical reaction, lactate oxidizes to pyruvate. Okay, but actually this enzyme, lactate dehydrogenase, like pyruvate reduction. So therefore, we can say this is a member of oxidoreductase. This enzyme catalyzes oxidation and reduction. And this is actual reaction. So if you reduce pyruvate, if you reduce something, you need to oxidize something. Okay. If you reduce pyruvate, you need to oxidize something. In this case, NADH. NADH is oxidized to NAD plus, coupling with pyruvate reduction. So this enzyme reduces pyruvate to lactate, and also they oxidize NADH to NAD plus. So therefore, oxidation and reduction is will be completed. Otherwise, you cannot do reduction only. You need oxidize too. Okay. So what is this? This is looks like this. NAD is nicotinamide, adenine dinucleotide. So you can see adenine here. This is adenine, and this structure is nicotinamide. This is a nicotinamide. So this compound name is just simple. Nicotinamide and adenine and dinucleotide. This structure we can sometimes say nucleotide, dinucleotide. This and this. Two nucleotides. Therefore dinucleotide. So simple this, this, and this. Nicotinamide, adenine, dinucleotide. So we have this kind of molecule in our body for to help oxidation and reduction. This is a chemical change, oxidation reduction of NAD and NADH. So just showing this structure. This is oxidation state. And if you reduce this compound, look like this. Okay. So we sometimes say this NAD is coenzyme. Coenzyme help the action of enzyme. So if you don't put NAD, the oxidoreductase cannot work, does not work. So this compound is essential for enzyme action. Okay. So again, this chemical reaction. So lactate dehydrogenase reduce pyruvate with NADH. Then pyruvate reduced to lactate, and NADH oxidize to NAD+. That's action of lactate dehydrogenase. So let's move to another story. This is the ethanol fermentation, ethanol production. So again, this is glycolysis from glucose to pyruvate, just same as our glycolysis. And this time, pyruvate is once decarboxylated. So pyruvate, this enzyme, pyruvate decarboxylase, this enzyme pull carbon dioxide from pyruvate, remove carbon dioxide from pyruvate, then produce acetaldehyde. Then acetaldehyde is reduced to ethanol. That's ethanol fermentation. Basically, glycolysis, but connected to ethanol production, like this. Removing carbon dioxide and reduction of acetaldehyde. So there are two additional enzymes for ethanol production. Pyruvate decarboxylase here, and alcohol dehydrogenase here. Okay, so this is the reaction catalyzed by pyruvate decarboxylase. Pyruvate here, and acetaldehyde and carbon dioxide. So this enzyme is very important for ethanol production. Pyruvate decarboxylase is not oxidoreductase. It's a group. It's a member in a group lyase. Maybe you don't know the name of lyase. It's just a group of the enzyme. This group catalyze carbon bond cleavage. In this case, cleave here, cut this CC bond, cut here, and produce carbon dioxide from here. And this part is going to be acetaldehyde. Just look like a cut carbon bond. This is a group of layers. Sometimes these layers catalyze opposite direction, reverse direction. I mean, making a carbon bond. Maybe this reaction cannot do a reverse reaction. I mean, carbon dioxide and acetaldehyde does not produce pyruvate, but sometimes this group of lyase enzyme catalyze carbon bond synthesis, making a carbon-carbon bond. And the important thing is by means other than hydrolysis or oxidation. So just this is categorize the enzyme. If enzyme catalyze hydrolysis or oxidation, it should be belong to other group. So therefore, this is a kind of restriction. The grouping of lyase. Lyase just making or cleavage of carbon bond like this. So anyhow, this pyruvate decarboxylase catalyze this reaction and produce acetaldehyde and carbon dioxide from pyruvate. This is a unique enzyme. Actually, we don't have this enzyme. The human beings does not have this enzyme, and only plants and also microorganisms have this enzyme. And microorganism is also limited species of microorganism possess this enzyme, like yeasts or limited species of bacteria. Yeah. This is an important enzyme for ethanol production. Okay, and also if you produce acetaldehyde from pyruvate by removing carbon dioxide, this is acetaldehyde. Acetaldehyde should be reduced into ethanol like this. This part reduced like this, and this is ethanol. Again, acetaldehyde, if you reduce acetaldehyde, you have to use NADH. So acetaldehyde reduced to ethanol, and NADH is oxidized to NAD+. So this is very similar to lactic acid fermentation, lactic acid production. So ethanol production needs two enzymes, pyruvate decarboxylase and ethanol dehydrogenase. Okay. So third example. So first one is lactic acid, second one is ethanol, and third one is amino acid. So amino acid also, so for example, I think you can see the amino acid in the supermarket in your country, but maybe you didn't noticed this is amino acid, but maybe you can find amino acid in your supermarket. So, glutamate. Glutamate is a very famous flavor enhancer. So maybe if you cannot see the glutamate in marketplace, but some foods contain synthetic, fermented industrial glutamate as a food additive. Very big market in the world. I'm not sure how big is, but a million tons in worldwide production. So I like to talk about glutamate production by microorganism, and also lysine. Lysine is also very high demand in the world for food for animals. Very big market in the world. So glutamic acid or glutamate production by Corynebacterium is one species of microorganism, actually. So this microorganism produce glutamic acid, glutamate from glucose, and we need to put ammonium ion, because glucose does not contain nitrogen. So this is, alpha keto glutarate in TCA cycle. It's one of compound in TCA cycle, alpha keto glutarate. This is alpha keto glutarate. That means this reaction is also basically metabolism in our body, not actually in our body, but usual standard metabolism in this microorganism. Microorganism produce alpha ketoglutarate as standard metabolism of glucose, and this organism produce glutamic acid from alpha ketoglutarate by using this enzyme reaction. Alpha ketoglutarate, ammonium ion, and NADPH. Again, this reaction is reduction of alpha ketoglutarate, and also put this ammonium ion here, this side. Then produce this oxygen removing to produce water, and this ammonium ion integrated in alpha ketoglutarate like this. Then produce glutamate. And this reaction is reduction. Therefore, NADPH oxidized to NADP plus. So this reaction, including addition of this ammonium ion into this compound, but basically oxidation reduction. So therefore, this enzyme, glutamate dehydrogenase, and this is a member of oxidoreductase. Okay. This is another example for catalysis. So I'm going to I'm going to talk about next important thing important factor of enzyme in fermentation. It's regulation. But maybe I don't have much time to talk about this second topic of enzyme function. So therefore, I like to move this topic to our next lecture. Okay, let's summarize my talk today. We discussed three questions, actually. The first one is what is enzyme? And enzyme is, sorry, this is a opposite. I'm sorry. So what is fermentation? Is the first question. And my definition for fermentation is look like this. Fermentation is chemical changes in foods with the aid of microorganisms. That's a kind of important factor of fermentation with the aid of microorganisms which are beneficial to humans. This is fermentation. And second is what is enzyme? So enzyme is biocatalysis, which accelerate chemical reactions related to biological processes. This is an enzyme. And what is the key of enzyme in fermentation? Of course, catalysis. This is very, very important. As I said in the pyruvate decarboxylase, we don't have pyruvate decarboxylase. So therefore, we cannot produce ethanol by itself. And so catalysis is really important. If you don't have specific catalysis, you cannot produce specific chemical compound. It's obvious. So catalysis is very important. But another issue is regulation. If you produce lots of compound like ethanol or glutamic acid or something, the regulation is very important. This another interesting issue to consider enzyme function. Okay, that's it, my talk today.
Okay, thank you, Professor Yakushi, for your talk. Before I open the discussion, okay, I will tell you that you can ask from by chat through the Zoom meeting, or you can ask directly. So maybe I can open the discussion, Sensei.
Right.
If you have a question, please ask to Professor Yakushi. Is there any question? Okay. Hadi Kurnia.
Okay, thank you for the time. I want to ask Mr. Yakushi, is there any way that we can see the fermentation? So you talk about fermentation for this and that compound. Is there any way we can see the fermentation process? And if yes, how can we see it? And if no, how can the scientists discover the pathway of the fermentation, since they can't see the fermentation process?
What do you mean? how can I see the fermentation? So, for example, in case of ethanol, alcohol fermentation, in that case, just we detect ethanol. Then we can recognize this is ethanol fermentation, things like that. This is your question?
I mean, the question is, how can we study the pathway here, since we cannot see the fermentation process, like from pyruvate to ethanol there is acetaldehyde. Can we see the acetaldehyde compound in real life or in real image?
So your question is, how do we study the pathway, right?
Yes.
It's for example, okay, if in the case of ethanol production, ethanol fermentation, we know the fermentation product is ethanol, right? And we consider which compound can be intermediate or can be intermediate to produce ethanol. So if you think about for example, if the acetaldehyde can be a candidate for intermediate for ethanol production, we can try acetaldehyde change. We can try whether acetaldehyde can be converted to ethanol by enzyme reaction. For example, enzyme reaction by using cell extract. If you prepare cell extract and put acetaldehyde into the cell extract, then wait for production of ethanol. Then if cell extract produce ethanol, that means acetaldehyde is the precursor for ethanol production. Then repeat this kind of experiment. How do they produce acetaldehyde? Then if you find carbon dioxide, when cell extract produce acetaldehyde, then we can consider this chemical reaction. Pyruvate to carbon dioxide acetaldehyde. Then, and also we can follow this enzyme activity by kind of fractionation experiment. Fractionation experiment leads to purification of enzyme. We can purify the enzyme by fractionation of cell-free extract, cell extract. Then define this enzyme actually catalyzes this reaction, things like that. Then we can determine the catalysis, chemical reaction in the fermentation, and also we can determine which enzyme, which protein catalyze this chemical reaction. Things like that.
How about Ms. Radi? Can you catch the explanation from Professor Yakushi?
Yes, I can get the explanation. So we need to take several procedure of reaction and to make to us to see the acetaldehyde and ethanol.
Right, right.
Yes. Okay, the second question is come from Rizkania. Please, Rizkania.
Thank you, Pak Nur. Good morning, Professor Yakushi. I would
Morning.
like to ask about the difference between lyase and pyruvate decarboxylase. Are they the same, or are they working together to perform acetaldehyde? Thank you.
Maybe my explanation was not good. Lyase is just a group of enzyme. How do I say? We can divide enzyme, all of enzyme into 6 group. So one of them oxidoreductase and lyase and anyhow 6 group. Then pyruvate decarboxylase is one of member of lyase group. Okay.
Okay. Yeah, I see. Thank you so much, Professor Yakushi. Probably I missed it earlier.
Thank you.
We still have 7 minutes. So if you have any question, please. No one?
May I have question, Pak Polis?
Ya, Bu Agustin, please.
Mahasiswanya nggak ada yang tanya. Thank you, Professor Yakushi.
Thank you.
This is a good presentation, so we can get many information from you. So I'm interested in amino acid production that you said. In amino acid production, you use biotin to regulate the enzyme activity in this case. Is it right?
I didn't touch actually, but
okay.
It's right. That's
okay.
right.
So actually, I would like to know about the why we use biotin in this case, so we can. Yeah, I'm interested in amino acid production.
Okay. Yeah, it's actually it's another interesting story in glutamic acid production by chorionet bacterium.
Yeah.
It's how do I say? Maybe it's found accidentally in the fermentation process. If you limit biotin, it's a kind of vitamin for this microorganism. So they need biotin because chorionet bacterium cannot produce biotin itself. So you need to put biotin into the growth media. But if you limit biotin, I mean, if you put biotin in a very low amount, in that case, this microorganism produce many glutamic.
Glutamic, yeah.
Yeah, that's a story. Then the molecular mechanism is kind of difficult to understand, but there are 2 things. One is biotin is related to fatty acid production. So if you limit biotin, maybe, of course, growth is limited, and also membrane is somehow weak, very, how do I say, weak, yeah, fragile membrane.
Fragile.
Very, easy to degrade. And also inhibit this enzyme, alpha ketoglutarate dehydrogenase. It's a required for usual TCA cycle. And if you inhibit this enzyme, the alpha ketoglutarate should be increased, then easy to redirect this compound to glutamate. So maybe the biotin limitation makes two things. One is repeat, and another is inhibition of this enzyme. Therefore, increase glutamate.
Okay.
Yeah.
Thank you very much.
Welcome.
Maybe this will be the last question for this guest lecture today. Is there any question? No question?
May I have one more, Pak colleague?
Yeah.
Okay. So you said also that the enzyme is not always protein, right? But you said this is also RNA. Could you explain about this? For example, what kind of the RNA or the enzyme?
It's difficult to explain the catalysis of ribozyme. It's a growing peptide in the ribosome. It's kind of なんだったっけ transpeptidase. Growing peptide connected to transfer RNA and coming amino acylated transfer RNA making transition of peptide bond.
Okay.
So peptide bond elongated by one. This catalysis is RNA, not protein. I cannot explain without any figures, but RNA molecule itself catalyzes this elongation of peptide.
Okay.
Yeah.
Okay. Thank you.
Thank you.
Thank you very much.
Okay, because the time is limited, I would like to say thank you on behalf of Head of Department of Agricultural Product Technology, because probably Dr. Widya Dwirukmi cannot attend this guest lecture. So on behalf of her, I would like to say thank you once again to Professor Toshiharu Yakushi, because you can give a lecture today. Maybe we will have another guest lecture on Friday and also next Monday.
Yes.
Okay, thank you very much also for all the participants for coming and join in this online guest lecture. Thank you very much, and see you next guest lecture.
Thank you.
Thank you.
Thank you very much, Professor Yakushi.
Thank you very much, Pak Nur and Professor Yakushi.
Thank you, Professor.